Thank you. Thanks for the invitation and for organizing this. I'm really looking forward to presenting this work. So I just started as a group leader in Dresden, but this is work I still did in my postdoc in Santa Barbara with Nicolas Clausen, really like an outstanding PhD student who's now at Princeton, and my mentor there, Boris Schreiman. And in my own group, my first PhD student has just started 2 weeks ago, so can't tell you anything about the new stuff yet, unfortunately. So in general, my driving interest is to understand development from going from an oocyte to an adult organism. And of course, biologists have studied in great detail the genetic patterns, for instance, here in the fruit fly, that sort of shape the body plan. But to get actual physical shape, forces have to act, growth has to happen, and so on, to generate physical form. And this is not a feed-forward process acting just through the molecular motors and adhesion proteins, but there's lots of feedback loops in this where mechanics and geometry feedback into the genes, and that's really where sort of the interesting physics starts to come in. And I like to think about tissue as sort of a material that shapes itself through collective dynamics and through these feedback loops. So just very briefly, the sort of two main directions of my research are, on the one hand, the sort of feedback-controlled tissue mechanics, and that's also what I'm going to talk about today, not just in animal tissues, but also, just to flash it briefly, in plant tissues, which are mechanically really interesting. And then on the other hand, it's sort of understanding pattern formation, so more in the traditional sense of reaction diffusion systems, but now coupled to things like orientational order parameters and also, again, tissue mechanics. And so I've done some work on Hydra that's published, and we're continuing along these lines, basically now coupling the mechanics side and the pattern formation side. All right, so let's dive into the specific setting for this talk, and this is Drosophila germ band development. And so this is a very early Drosophila embryo that you see here, and this is experimental data, microscopy data from cell membranes that have been labeled, that is computationally processed. So these images, microscopy images, have been segmented and tracked and false color coded so that you can sort of follow the tissue movements as I now run this as a movie. So in the top, you see the original embryo geometry, and on the bottom, you see sort of a map projection with sort of the characteristic distortions near the pole that allows you to see the entire half of the embryo. During this process called germ bed extension, where this blue tissue here extends by a factor of 2 along the body axis. I'm going to run the movie again, and compresses along the back to belly axis, the dorsal ventral axis of the embryo at the same time. And thanks to this super nice microscopy and segmentation pipeline, we can follow this process not just on the tissue scale, but we can resolve all the cell scale processes that underlie this. So we can distinguish in particular between cell shape changes and cell rearrangements that together drive the tissue strain. And let me just emphasize here, there's no cell divisions happening during this process. So these are the cell shape changes, and rearrangements are the only ones that contribute. And this is a two-dimensional sheet of tissues, an epithelial sheet that floats on a yolk, essentially. And so really what's happening is all in the plane, and the cells are exerting forces on one another. They're not crawling on a substrate. When we zoom in onto these cell rearrangements, we can see that they're really highly organized, as you can see from the sequence of snapshots down here. And we would like to understand, how do the cells do this? How do they generate this tissue scale flow from these cell scale processes? And sort of asking this question from a physics perspective is that we, on the one hand, see a tissue flow that appears fluid-like when we squint our eyes, but when we think about morphogenesis, where a permanent sort of resilient shape has to be generated, this is something we would associate with a solid that can resist deformations and after a perturbation can return to its target shape. And so the question is, how does the tissue bridge these two different rheological behaviors? So to briefly recap conventional elasticity, there are two main ingredients. One is that there is a reference configuration, and then the deformation of that reference configuration to the physical configuration that we observe defines a strain tensor, So this is this U alpha beta here. And then from the strain tensor, we can calculate the stress via a constitutive relation. So this is the material law that describes basically the material properties. And then once we have these two ingredients, we can solve the mechanical problem. So let's see how these things work out in a tissue. So in an epithelium, the dominant forces are generated along the cell-cell interfaces where two cortices of the two cells meet, and they have actin and myosin generating tension and basically sort of these contractile forces. And these can be very nicely visualized when one does a laser ablation experiment where one takes a laser and cuts one of these cell-cell interfaces and then can see the recoil, which happens very rapidly compared to the timescale of morphogenesis, and which demonstrates that all of these cell-cell interfaces are under tension, which is generated by the myosin motors. And because the tension is generated by the myosin motors, it's actually independent of strain. Just as I can tense my muscle when it's extended and also tense it when it's short, in the same way, these junctions can generate tension under different length. And so that means, on the one hand, because of molecular turnover, there's no reference configuration. And then also because of this decoupling of stress and strain, there's no constitutive relation. So we're losing both elementary ingredients of elasticity theory. And so the only thing that we're left with, but that's going to rescue us, as you will see in this talk, is that there is a timescale separation between fast mechanical relaxation towards force balance compared to the slow morphogenetic process. So that's what we're going to rely on throughout the talk. So to give you a basic intuition for how something that behaves like an elastic solid can emerge from a microscopically fluid material, you can take a glass of soapy water and you can make soap bubbles out of it. And even though the surface of the soap bubble is a fluid film, with a surface tension, it behaves like a solid in the sense that the soap bubble has a defined shape. It has a defined radius, and if you poke a soap bubble, you deform it a little bit, it will spring back to its spherical shape. And then indeed, if you take many soap bubbles and assemble them into a foam, then you can make little sculptures out of the foam and they will hold their shape against gravity. So a foam is actually an emergent solid. It's microscopically made out of a fluid, but macroscopically has the properties of a solid. Of course, with a very low yield stress, so if you shear the foam too much, then it will permanently change its shape through plasticity. And so throughout the rest of the talk, I'm going to take this intuition and transfer it to the setting of tissues. So here's the way one formulates this mathematically. So we can write down a virtual work function that basically describes the virtual work that's generated through length changes under tension of the junctions and through area changes against pressure. And here we're going to assume that pressure is identical in all the cells. And I'm happy to discuss afterwards how we can relax this assumption. So let me just very briefly, for people that are familiar with sort of these more canonical vertex models, emphasize that this is quite different from the vertex model you might be familiar with, which has this sort of area and perimeter elasticity as written in the gray box here, because we're saying that there's no constitutive relation, there's no which would be the Kp and Ka here, they're like a constitutive relation. There's no reference configuration. So in the vertex model, P naught and A naught act like reference configurations. We're saying we're prescribing the tensions. And we're trying to figure out how the tissue mechanics emerges from that. So it's not that, right? And instead, we're going to rely on force balance. So this is just taking the variation of this virtual work with respect to the positions. You end up with a force balance equation that just says that the forces due to tensions on each of these interfaces, they have to add to 0 at a vertex. And so we can geometrically represent this as a triangle because three vectors that add to 0 form a triangle. And if we rotate this triangle by 90 degrees, then each red edge, each triangle edge is perpendicular by construction to the corresponding cell-cell interface. And that means that now the angle at the vertex here will be complementary to the angle in the triangle, meaning they add to 180 degrees. So that's basically the geometric link between the triangle and the polygonal cell tiling. And now we can do this at all the vertices, and that means now we have a triangulation because neighboring vertices, they share one interface in between, and the tension on that interface is represented by the corresponding triangle edge. And so neighboring triangles have to share these edges. And the length of each triangle edge represents the level of tension on the corresponding cell-cell interface. So let me very briefly show how we can use this machinery to infer the tensions from the observed geometry. So we can take the microscopy images that originally look something like this, and then segment out all the cells, and now take this link between the geometry and the tensions to infer the relative tension on each interface. And so this is color-coded here, where gray means a relative tension of 1, so it's identical to the neighbors as it would be in a conventional fluid foam, and then you would have 120-degree angles at each vertex. And red means higher tension compared to the neighbors, and blue means lower tension. And so we can do this for an entire movie from the experiment and observe how these tensions evolve over time. And in the particular example of this Drosophila early ectoderm, we see this alternating pattern of high tension, low tension, high tension, low tension emerge, which if we now focus on a quartet of cells with one of these high tension interfaces in the middle, then these high tensions will continue to increase as the junctions shrink, as the interface shrinks. And that will give rise transiently to a fourfold vertex, and then a new interface emerges. And this is called the T1 process, and this is happening all over this piece of tissue and drives the elongation of that tissue. So that's what's underlying convergent extension, so this sort of elongation flow of the Drosophila tissue during early embryogenesis. Let me run this movie again so you can see that all these high tension interfaces contract, undergo the T1 processes, and as that happens, this nice alternating high-low pattern that we saw earlier gets destroyed, and this actually leads to stalling of the tissue flow. And at the very end of the talk, I'm going to come back to this point where I'm going to present a model that exactly reproduces this kind of behavior. And let me also maybe briefly flash that we can, of course, not just make nice movies with this, but we can actually quantify systematically the dynamics during these T1s and so on. And that's going to inform the model later in the talk. So now let's get back to this idea of stress-first mechanics. So instead of inferring the tensions from geometry, let's ask the questions, if I give you all the tensions between the cells on the cell interfaces, can you tell me what the geometry of the tissue will be? And so here's what this looks like geometrically. So the tensions are encoded in this triangulation. So the first thing we have to do is we have to take all the tensions which specify the edge length in the triangulation, and we have to embed these in the plane to get the actual triangulation, to basically get the vertex positions here labeled with these taws, with these bold taws, in the plane. So once we've done that, that's basically just sort of like a layout problem. We have to construct the polygonal cell tiling. And the way we do this is by using Delaunay-Voronoi duality. So for those who don't know this, the way that a Voronoi tiling is constructed is by taking the circumcenter of each triangle, so the center of the circle that touches the three vertices, here shown for instance, these two green circles, and then connect the circumcenters. So connect these with an edge. And if you do this for all the triangles and their neighbors, then you get a polygonal tiling that has exactly the property that we need to ensure force balance. And that is that the triangle edges meet the cell edges, the cell interfaces, at a 90-degree angle. Right? Remember, this was the defining property of the triangulation, that we took the original tension vectors and we formed a triangle and rotated it by 90 degrees. So that's the geometric link between the triangulation and the polygonal tiling that corresponds to this force balance constraint. And now we can turn this Voronoi tiling, which at the moment is just sort of an arbitrary choice of geometry, into an actual physical object by asking, well, what is the stress tensor on a microscopic scale that corresponds to this Voronoi tiling? And so the way we calculate the stress tensor is by basically imagining that each cell-cell interface is a little force dipole, and we're taking the average over all the contributions from these force dipoles over some area of tissue. And this is sometimes called the Batchelor stress formula or virial stress formula. And if we evaluate this formula for the Voronoi tiling, then what we find is quite remarkably that the stress tensor is isotropic and uniform. And that means it's trivially balanced by a global pressure, which of course has to be there to prevent the whole thing from collapsing. So the Voronoi configuration is like a stress-free reference configuration, right? Because it's trivially balanced by a global pressure. The tension is trivially balanced everywhere. And that reference configuration, I really have to emphasize, really here emerges from the prescribed tensions, which was the starting point for this construction. Now we can take a next step and we can ask, well, is this the only configuration that's compatible with force balance? And it turns out it's not, because again, force balance constraint is just this perpendicular angle condition between the triangle edges and the cell interfaces. And we can actually move around the cell interfaces without changing any of the angles. So there are two examples of this here, where for simplicity, all the angles here are 120-degree angles, like they would be in a conventional fluid foam. But obviously, the length of the interfaces are different, and so the cell shapes are different, and the tissue shape overall is also different. And this means that mathematically, we can represent these kind of deformations as irrotational displacements of the vertices. These are displacements that change the length, but do not change the angles. And we can represent irrotational displacements as displacements that derive from a discrete gradient, here called theta. So there's a scalar quantity in each cell, a theta I, a theta j, and so on, that parameterizes this set of deformations, that preserves the angles and therefore preserves force balance for the same set of tensions. Now, again, we can calculate the stress tensor with this virial stress formula. And what we find, I'm not going to show the full expression here for simplicity, but you can imagine it. What comes out of this is because there's this factor L, this edge length factor that comes in here, that the macroscopic stress tensor is proportional to the cell strain. So for instance, for the example on the bottom here, where all the cells are a little bit stretched along the vertical, the stress tensor would have a contribution that is higher along the vertical than along the horizontal. And now we have a stress that's proportional to strain, and that means we have emergent elasticity, because we have something that looks like a constitutive relation that emerges from this geometric construction. And we can actually take the continuum limit of this quite easily. So this gradient very easily translates into continuum, and what comes out is that this theta indeed plays the role of the Airy stress function, which guarantees force balance in elasticity. So any stress tensor that derives from an Airy stress function is automatically in force balance. Now, to sort of emphasize this point again, what we started with were interfacial tensions on the cells. And experimentally, you can appreciate these from these, or measure these from these laser cutting experiments. And these are what defines the cell geometry, but it leaves these extra degrees of freedom open. And these extra degrees of freedom exactly correspond to force-balanced macroscopic stress tensors on the tissue scale. And so these are experimentally measured by doing laser ablation, but now on a larger scale, where you cut, for instance, a circle in a tissue where the circle contains maybe 100 cells, and then you can watch how the shape of that circle after cutting relaxes and recoils. And that's measuring tissue scale stresses. And the important insight here is that really there's a key difference between the cell scale tensions and these tissue scale stresses. They are completely different objects. And indeed, that's again familiar from a foam, where in the fluid foam, all the interfacial tensions are just a material parameter. It's just surface tension. But then the macroscopic stress in the foam is determined by external forces that are acting on it. And the same situation is true in a tissue. Now, what's fundamentally different in a tissue from a foam is that in the tissue, cells can generate tension actively, and they can decide how much myosin is loaded onto each interface, and so they can dynamically adjust these tensions. And so this is how we go now from a statics problem to dynamics through the changes of the tensions. So this is a reminder that's sort of the mathematical setup for the statics problem. And now geometrically we have represented this through this triangulation framework. What a dynamic change of tension means is that the triangulation changes its shape. And therefore, the stress-free reference configuration that we constructed with the Voronoi tiling is also going to change. And then finally, of course, there are external forces that act on this, which eventually give the physical state of the tissue. But the key here really is that dynamically, through changes of the tensions, the physical shape can change even in the absence of the external forces. So to have a specific model for this, we need to decide basically how do these tensions now evolve over time. And inspired by these results that I showed you earlier, where we saw that tension increases nonlinearly on the interfaces that undergo these T1 processes, we propose a very simple model where basically we say that each tension reinforces itself. So there's some positive feedback with an exponent n here that's driving up the tension. And at the same time, because there's a limited pool of myosin in each cell, when tension on one interface increases, it necessarily decreases tension on the neighbors. So there's basically a winner-takes-all mechanism in this positive feedback. And that's now driving, when we simulate this is driving T1 processes, where if you start with one interface that has slightly higher tension than its neighbors, initially it's going to increase in tension and it's going to drive the neighbor exchange, like you see in this little simulation here. And we can now take this local rule, this local feedback rule, and implement it on an entire tissue, which in tension space is represented again through this triangulation. This is where the local feedback happens. And then through force balance, this defines the corresponding physical configuration of the cells. So this is the one shown down here. And I'm going to initialize these simulations with a slight anisotropy of tension along the vertical. So the tension is slightly higher there than on the horizontal interfaces. And I'm comparing two initial conditions. One where the cells are all hexagons, so there's topological order, and one where there's topological disorder. So there are pentagons and heptagons and so on. So now when I run these simulations, you're going to see that even when I start from order, due to these T1 transitions, these neighbor exchanges, the tissue becomes increasingly disordered, whereas the disordered tissue just stays disordered. And the initially ordered tissue manages to undergo a larger amount of strain, of sort of shear flow, than the initially disordered tissue. But eventually both of them become highly disordered, and in both of them the flow stalls. And that's exactly what we see in Drosophila. So in Drosophila we see that there's a fast phase of tissue extension here showing the aspect ratio of a patch of tissue. And then as this aspect ratio change reaches a factor 2, then the tissue flow slows down and the tissue becomes increasingly disordered. So this model with this very simple local feedback rule reproduces what we observe in Drosophila, and the key principle underlying this is really that there's this tight link between the dynamics of tensions and the physical geometry that is enforced by the force balance constraint. So to summarize, I presented a framework where we think about epithelial tissue as basically a distributed hydrodynamic skeleton. So there's this emergent elasticity that comes out of that. And we can represent this geometrically with this triangulation. And that dynamics happens not through an imbalance of stress, of active stress that is balanced by viscous dissipation, but through this adiabatic of the microscopic tensions, which then, through this emergent elasticity, give rise to tissue scale shape change. And finally, sort of zooming out to a more biological picture, this is showing that there's local self-organization. There's a simple local positive feedback that is able to drive a global tissue flow. So to briefly give an outlook, we have recently taken this framework from a discrete level and managed actually to derive a very nice continuum limit from it. which relies heavily on the mathematical correspondence between triangulations and Riemannian manifolds. And we are also now thinking about generalizing this idea of shape being controlled by stress in force balance to three-dimensional tissue shapes. So in a very simple setting for mouse blastocysts, it has been shown that the radius is controlled by exactly Laplace law, basically by the balance of tension and pressure. And we're asking, can we use this idea and generalize it to more complex shapes, like this shape from the Drosophila Midgard from my collaborator Noah Mitchell at U Chicago, where it could also be a stress pattern in the surface that's controlling the three-dimensional tissue shape. With that, let me thank you for your attention. I'm looking forward to questions.
Thank you, Fridtjof, for a wonderful talk. We already have some questions in the chat that I'm going to ask. The first question is from our organizer, Jing Chen. Jing is asking this is from early part of your talk. How does the strain tensor characterize the remodeling of cell-cell adhesions?
Oh, okay. Let me unpack that. So let's go to maybe this. So adhesions really come in here as you can think of it as modulation, as also a way as an ingredient that modulates the tensile stresses. So often people like to think about adhesion as counteracting tension, like basically like a negative surface tension. I'm not sure if this is quite the right way to think about the role of E-cadherin in this. But certainly adhesion on a more basic level is needed to just keep the cells together, right? Without adhesion, the tissue wouldn't be confluent, and then this whole thing, like an epithelium, would just fall apart. To link it to the cell shape, since this was also part of the question, the cell shape really comes in through the length here, and of course through these e's, the unit vectors along each of these edges. Which encodes indirectly the cell shape, and then also through this Batchelor formula gives rise to the stress tensor. So that's where the emergent elasticity comes from. I hope this roughly answered the question.
Thank you. And we can take more, discuss this more during the discussion. So the second question is from Column, and Column is asking, how close are experimentally observed cell tilings to Voronoi tessellations?
Yeah, okay, so excellent question. So the Voronoi tiling here shows up as the stress-free reference configuration. And so when you see a Voronoi, then that tells you that microscopically this tissue is stress-free. What we see in practice is that there are always some deviations which we can then exactly link to the sort of macroscopic stresses. So we can basically measure, or at least infer, passive elastic stresses in a tissue by measuring the deviation from Voronoi.
Thank you. Okay, We'll take one more questions now. So which is from Emily, and she's asking, how do you manage 4-cell vertices and rosettes in this triangulation formulation?
Okay, great. So I have, let me skip to a backup slide for this. If I can find it quickly, here it is. Okay, so the construction with this Voronoi construction has a very nice property, and that is the occurrence of a fourfold vertex is exactly then when in the corresponding triangulation, when the two triangles that oppose this edge. When these angles add to 180 degrees, the angles opposite of the interface, then that interface length reaches 0. And so there's a very immediate geometric condition for when T1 transitions have to happen that you can read off from the triangulation directly. And that's actually what we exploit in the simulations, where we don't even construct the Voronoi sort of during each simulation step, but we just simply take that geometric condition in the triangulation because we know this is exactly the point when a T1 has to happen, because that's when a fourfold vertex comes in. Higher fold vertices like rosettes would basically correspond to situations where you have to generalize this kind of construction and where multiple pairs of triangles that are all next to each other all have this property that angles add to 180 degrees.
Thank you, Fridtjof. Thank you again for a wonderful talk. There are more questions for you, which we will take during the 15-minute discussion.